In this talk, we're going to learn about taxonomy, systematics, and phylogeny. There is immense biodiversity on Earth. We can see many different types of organisms, and these organisms come in a variety of sizes and shapes. In order to study them and learn about them, the best way to accomplish this is to classify them into groups. Taxonomy is the method of identifying and classifying organisms into groups. The most widely used method of classification was established by the scientist Carl Linnaeus, and hence it is called as the Linnaean classification. The Linnaean classification is based on different factors and characteristics that are present in a variety of organisms. These include the phenotype displayed by different organisms, their morphology, the anatomical features, as well as physiological features. The Linnaean method of classification involves the creation of different groups that are arranged in a hierarchy. Each group is called as a taxon, and the plural of taxon is taxa. Let us learn about these different groups in the context of the American black bear. The American black bear is the common name of the animal, but please note that every organism has a scientific name as well. This scientific name is based on this hierarchical method of classification. So let's have a look at this method. The first group or taxon is the domain. The American black bear resides in the domain of Eukarya because it is a eukaryote. The next taxon is the kingdom, and because the American black bear is an animal, it belongs to the kingdom of Animalia. The next taxon is phylum, and since the American black bear is a chordate, it belongs to the phylum of Chordata. The next taxon is the subphylum, which is not shown in this figure. But in the case of the American black bear, it belongs to the subphylum of Vertebrata because it has a vertebral column. The next taxon is the class, and the black bear is in the class of Mammalia. The next taxon is the order, and the American black bear is in the order of Carnivora. The next taxon is family, and the American black bear belongs to the family of Ursidae. Following the family comes the taxon of genus. The American black bear is in the genus of Ursus. The last taxon is species. The American black bear belongs to the species of Ursus americanus, and it should be noted that in the case of species, we do include the name of the genus as well. Thus, the scientific name of the American black bear is Ursus americanus. As mentioned before, all organisms have a scientific name which is based on the Linnaean classification. The naming system is called as binomial nomenclature because the name comprises of two words. The two words refer to the genus and the species that the organism is placed in. There are certain rules of binomial nomenclature. These include that the first word of the name of the organism is the name of the genus. The next word, or the second word, is going to be the name of the species. So in the case of the American black bear, the binomial nomenclature states that its scientific name is Ursus americanus. Another rule to keep in mind is while writing its name, the first alphabet of the name, which is the name of the genus, has to start with an uppercase alphabet. While typing the name, we should always italicize the name, but while writing the name by hand, we always underline the name. These are certain rules to keep in mind whenever we are writing the scientific names of different organisms. Taxonomy is a great way to group organisms and learn about them. However, it is not a reliable indicator of evolutionary relationships. For example, let's look at chimpanzees and humans. Taxonomy does tell us that both are in the class of Mammalia and hence are mammals, but it does not tell us whether they share a direct ancestor or a distant ancestor. We know that all living organisms that are there today arose from ancestors that were there in the past. And thus, the taxonomy does not tell us any information on ancestry. Here's another question. What about humans and lizards? They are in different classes, but do they share ancestors? To answer these types of questions, the field of systematics plays a more important role. It is the study of diversity and evolutionary relationships among organisms. The field of studying evolutionary relationships is also called as phylogeny. The evolutionary relationships are depicted using these pictorial representations called as phylogenetic trees. Phylogeny comes from the word philo, that means tribe, and geny, that means generation. Thus, phylogeny is the generation of groups that allows us to classify organisms into tribes, which are organisms that show similar characteristics. Whenever we set up a phylogenetic tree, it can include not only the presently living organisms, which are called as the extant organisms, it can also include extinct organisms, which are organisms that lived many years ago but are no longer living because they became extinct. Let us now learn how to read a phylogenetic tree. A phylogenetic tree is made up of branches and nodes. The nodes denote different ancestors. These ancestors can be recent or distant. Let's take the example of chimps and humans. As we can see, they arose from the same direct ancestor. On the other hand, when we look at lizards, for example, lizards arose from an ancestor that is not the direct ancestor of chimps and humans. And hence, the ancestor that gave rise to lizards is a more distant ancestor. This way, we can keep going further and further away from the groups of chimps and humans, and ultimately we are able to see the starting node, which denotes a distant ancestor, or the first ancestor through which all these different groups shown in the tree have evolved. Let us now look at the branches of a phylogenetic tree. The branches are represented by the lines, and each branch is a hypothesis, which is basically a prediction of the evolutionary relationship of the different organisms to each other. The location of an organism can be revised and changed based on the information that is available. And that is why we say that each branch of a phylogenetic tree is a representative hypothesis, because we don't know with absolute certainty that its location that has been shown is indeed the right location. Now, organisms that are at the end of each branch are usually representative species. Now, species, as we saw, is a taxonomic group. In the case of a phylogenetic tree, sister taxa are those species that are descendants that arise from a node. So in this case, we are able to see that chimps and humans arose from the same direct ancestor and hence are considered to be sister taxa. Phylogenetic trees also show the presence of outgroups. Outgroups are taxa that are closely related to a group of interest, but they diverged earlier. So let us take the example of the sister taxa of chimps and humans. They arose from a common direct ancestor. Now, when we look at the lizard group, the lizards arose from an ancestor that is more distant than chimps and humans. And hence, in this case, the lizards are considered to be the outgroup. Similarly, if our group of interest is the group that includes the frogs, the lizards, the chimps, and the humans, then in this situation, the group of fish serve to be the outgroup because they did not arise from the same ancestor that gave rise to the frogs and the lizards and the chimps and the humans. The outgroup allows to root a tree. And in this instance, the root is going to be the part of the tree that shows the last common ancestor of all the taxa that we are interested in, which in this case were the frogs, the lizards, the chimps, and the humans. There are different ways to view phylogenetic trees, and one way is what we have seen so far, which is a tree that has horizontal branches. We can, however, rotate the whole tree to give rise to a vertical tree, where in this case the branches are represented as vertical lines. Similarly, we can also have phylogenetic trees where the branches are represented as diagonal lines. In all cases, the tree is giving us the same information of ancestry. The branches of phylogenetic trees can also be rotated, and when this happens, it still shows the same evolutionary relationships. For example, if we go ahead and rotate the position of the chimps and humans, then we can end up with the phylogenetic tree where we can see that the humans are shown first and then the chimps later. When we do the same thing for lizards, we can now see that the lizards are now below the group of humans and chimps in this newly constructed phylogenetic tree. The same thing can be done with the group of fishes, and when they are rotated, they are now seen at the bottom. Despite all these rotations that have been done at various branches, the evolutionary relationships do not change. Linnaean-based classification may hint at evolutionary relationships among the different taxa, but it does not explicitly imply any phylogenetic relationships. Let us look at the example of different classes in the subphylum of vertebrata, which includes amphibia, reptilia, aves, and mammalia. If we had to construct a probable phylogenetic tree based on the taxonomic information, we would construct something like this, which shows that the class of amphibia, reptilia, aves, which are basically birds, and mammalia all arose from a direct common ancestor. However, based on information that is used to build phylogenetic trees, we can see that these classes do not share a direct common ancestor. On the other hand, we can see that the amphibians rose from an ancestor of bony fish or osteichthyes, and in turn, an ancestor that was amphibian in nature gave rise to the ancestors who are descendants of current-day reptiles and mammals. Similarly, we can see that a reptilian ancestor gave rise to the class of aves, which are basically birds. Hence, taxonomy is not indicative of evolutionary history. Many different organisms have been classified in their appropriate taxonomic groups, but in some cases, they have been classified in groups which they don't belong in because they're not as similar as the other members present in those groups. For example, the plant Alicelia pinnatifida was originally classified in the genus of Gilia, but now it has been reclassified into the genus of Aliceala. Hence, it begs the question, why did we classify organisms into groups that they do not belong to in the first place? One of the answers resides in the information that was used in order to classify organisms. The taxonomic classification was done before DNA or protein sequencing was easily available, and hence molecular data was usually not considered while classifying organisms. On the other hand, features such as morphological, anatomical, and physiological characters were used, and similarities based on these characters was used for taxonomic classification in the past. Homologous features were predominantly used for classification purposes. Homologous features, such as homologous structures, are structures that have different appearances as well as functions, but when we look at their anatomy, the anatomy is quite similar. So in this picture, we are looking at the forearm of different types of mammals, including humans, cats, bats, porpoises, and horses. We can all agree that the forearm of these different animals are used for different purposes. However, when we look at their anatomy, we can see that they are made of similar types of bones that are arranged in a similar fashion. Thus, these are homologous structures. Homologous structures indicate descent from a common ancestor, and thus all these different animals are showing the same arrangement of bones because they arose from a common ancestor that had this arrangement of bones in their forelimbs. Many times, similarities in external features have been used for classification purposes, but it should be noted that there are instances where these similarities occur due to convergent evolution. Convergent evolution is when distantly related organisms tend to show similar phenotypic appearances. Let's look at an example of these marine animals. They all have a streamlined body, and hence their body appearance is similar. However, 3 of these are fish, while the last one is the dolphin, which is a mammal. Hence, they evolved from different ancestors but show a similar type of physical appearance. This is because of the common environment of water in which they reside in, and hence, this streamlined body is an example of convergent evolution. Convergent evolution can also give rise to analogous structures, which are structures that have similar functions but different anatomical features. A very common example is the wing of a bird and a butterfly. The wing of the butterfly and the bird allows both of the organisms to fly. However, when we look at their anatomical features, the wing of the bird has bones that support the wing, but we don't see the presence of any bones in the butterfly. Thus, analogous structures, which are also called as homoplasies, are not indicative of common ancestry and thus should not be used to determine evolutionary relationships. Since genomes or DNA sequences are inherited from ancestors, scientists widely accept that genomes retain the evolutionary history of organisms. So, if we take the genes present in 2 organisms and try to align them to see how similar they are, and if they do share many regions in their nucleotide sequence, as shown in this figure, then these 2 genes are most likely homologous. And hence, we can conclude that the organisms are most likely related to one another. This is the basis of molecular phylogeny. On the other hand, if we have a scenario where the genes present in 2 organisms are not as similar to one another in their nucleotide sequence, then we can also conclude that most likely these 2 organisms are not closely related. any similarities in their nucleotide sequences that can be observed in the figure are purely coincidental, and this is an example of a molecular homoplasy. Homoplasies are similar features that are independent of descent, and hence homoplasies should not be taken into account while determining the evolutionary history of organisms. In addition to DNA, protein sequence can also be used to determine molecular phylogeny. Nowadays, both DNA and protein sequencing tools are much widely available than they were in the past, and hence it is easier to build phylogenetic relationships based on this molecular data. As more morphological, anatomical, physiological, as well as DNA and protein information is obtained, organisms are being reclassified into their appropriate groups based on their evolutionary history. The main goal right now is to create groups or taxa that include an ancestor and all of its descendants. Such groups that include the most recent ancestor and all of their descendants is called as a monophyletic group. An example of such a group is the class of Mammalia, which is the taxa that includes all mammals. In this case, we are able to see that all mammals descended from one common ancestor, and that ancestor gave rise to many different other ancestors that ultimately gave rise to all the different organisms that come under the class of Mammalia. Since this group includes the ancestor and all of its descendants, it is a monophyletic group. The goal right now is that all taxonomic groups should be monophyletic. These monophyletic groups, or taxon, are also called as clades. Not all taxa are monophyletic in nature. There are instances where we have a group that includes the most recent ancestor but not all of its descendants, and such groups are called paraphyletic groups. Let us take the example of the class Reptilia. When we look at the phylogenetic tree, we are able to see a common ancestor in a box, and we're able to see all the descendants. When we look at all of these descendants, only the ones in the shaded boxes come under the class of Reptilia. We are able to see a glaring exception of a descendant that is not included in this class, and that includes birds. The phylogenetic tree clearly shows that birds did descend from ancestors that gave rise to all other reptiles. However, they are still not included in the class of Reptilia. Thus, the class of Reptilia is a paraphyletic group because it does not include all the descendants that came from the most recent ancestor, and hence the class of Reptilia is not a clade. It should be noted that there is a lot of hesitation to include birds in the class of Reptilia because they have their own class, which is called as aves, and it may create a lot of confusion by trying to merge the class of aves with the class of Reptilia. Thus, a clade is a group that includes a common ancestor and all the descendants that arose from that ancestor. These descendants could be living or extinct. A phylogenetic tree that shows clades is called as a cladogram. Now, a cladogram can include only one species, or it can include more than one species, as shown in the figure. Additionally, a cladogram must include all the descendants so that it is a clade, and it must also include all the ancestors that gave rise to the different descendants. With this, we come to the end of our talk, where we learned about the Linnaean-based taxonomical classification and phylogenetic trees. We learned about the relationships between taxonomy and phylogeny, and learned about how molecular phylogeny is now more commonly used to determine evolutionary relationships. We also learned about monophyletic and paraphyletic groups and how monophyletic groups are called as clades. We then learned about cladograms.